Well, hello everyone. We have a sleepy little boy here. Just thought I would show you the sleepy little boy before the video. Say hi, Kaiser. Hi, Kaiser. Sleepy. He's going to be laying here while we do the video. Okay, so our new unit is going to be biotechnology, and this is a mini unit. We usually don't even have like a full test. We have a TIS on this unit. But this video is going to introduce you to biotechnology and what's in the unit. So biotechnology is essentially how we can use DNA and the tools of DNA to help us do things in our life. So if you take a look at some of the pictures that are here, you'll see here this is what they call gel electrophoresis. And gel electrophoresis can be used to identify criminals by the DNA found at a crime scene. So we will be doing a lab where we have sort of artificial DNA found at a crime scene, and we do this process called gel electrophoresis, which we're going to talk about as soon as we get back from Christmas break, that allows you to cut DNA, and cut DNA based on the sequence that the person has, and using that you can identify whose DNA is found at a crime scene or not. You can also see that we have in the here, bless you, Kaiser. We have here a glowing mice and a pig with a glowing snout. So we also can now insert genes into organisms to give them traits. So here, in both the pig and the mouse, we inserted green fluorescent protein into the mouse and then into the pig, and that makes them glow. We can also insert genes that are helpful. So here, this is rice, and we've inserted a gene that allows it to be more nutritious and make more. I think it's a vitamin E. It makes more of, so it's more nutritious. But we can insert genes into organisms that help humans. We did this a lot with genetically modifying plants to make them grow faster. But essentially, we can use our genes and insert genes into organisms to give them traits that we want. You'll also see up here there's a bottle of insulin. Insulin is given to diabetics because they don't have the ability to take in sugar to their they don't have the ability to produce insulin, which signals cells to take in sugar. And we actually now produce insulin by giving the human gene of insulin to bacteria, and bacteria produce it for us. As soon as we get back from break, after we talk about gel electrophoresis, we're going to talk about how we can use bacteria to produce genes or produce proteins that we need. And even in this lesson, we're going to get a little bit of an introduction to it. So essentially, what is biotechnology? Biotechnology is the way we can manipulate DNA and use it for things that are useful to us. In this lesson, we're going to learn about some tools of biotechnology. And in this lesson, we're going to learn about plasmids, which we talked a little bit about in the bacteria lesson that you should have watched first. And we're also going to talk a little bit about something called restriction enzymes. Another word for biotechnology could be genetic engineering. And genetic engineering is when you manipulate DNA so that you can use it for your own usage, or things that are beneficial to you and the human race. So we're going to start off with describing or explaining again what plasmids are. And so if you look at a bacteria cell, a bacteria cell has a circular chromosome, and then a lot of times they have other smaller circles of DNA. And those smaller circles of DNA are called plasmids, and they have the ability inside the bacteria to self-replicate. They usually carry between 2 to 30 genes, and sometimes, bad for us, they also give the bacteria genes for antibiotic resistance, which make them not be able to be killed by antibiotics. Bacteria also have the ability to exchange plasmids. You saw in the previous video that bacteria can get genetic variation by something called conjugation, which is when two bacteria essentially hook up through its sex pulse, and then through that sex pulse they can exchange these plasmids, and they can replicate the plasmids and then exchange them or give them to another bacteria through conjugation. Okay, and they can also be taken from the environment. If there are plasmids in the environment, we're going to see that bacteria have the ability to take up this naked DNA in the environment and bring it into their cell. We actually have been exposed to this to some extent back when we were learning the Griffith Experiment, which we just had a test on before Thanksgiving, before Christmas. And one thing that is true is that bacteria have the ability to take up naked DNA. And we saw this in the Griffith Experiment. Remember, Griffith had bacteria. This is heat killed S bacteria, and he also had regular R. And when he mixed them together, and normally heat killed S, when it was heat killed, couldn't kill, and R couldn't kill in general. But what he saw, what he concluded, if you remember from this experiment, is that something from heat killed S went into R to give it to the ability to kill, and then through all the other experiments, they found out that material that went from heat killed S into R was DNA, but the ability of bacteria to take up DNA from the environment, which is what this R bacteria did, is called transformation. Okay? So in transformation, bacteria incorporate DNA from the environment into their own chromosomes and express it. And that's called transformation. Also, the word recombination, which we're going to be genetic recombination, refers to when organisms take in genes from another organism and put it into their DNA. So in some ways, when viruses were injecting their DNA into bacteria and then putting their DNA into the genome, that was recombination. When DNA from two different sources is combined, that's recombination. So we're going to see in this unit that we can use plasmids to help us. Okay, plasmids give us the ability to insert genes that we want into bacteria and make the bacteria produce them. So if you take a look at this picture here, this is a plasmid. If we cut the plasmid and then cut the gene, like let's say this is the insulin gene that we want the bacteria to produce. If we cut the plasmid and cut the insulin gene, we can then insert the insulin gene into the plasmid. We're going to see how we do this, and then glue the DNA. That would be done by DNA ligase. And then we can force, and we're going to see how we do that in a lab, we can force the bacteria to take in the plasmid. And then because the bacteria is stupid, it's going to transcribe and translate this plasmid, and then we're going to get, in this case, if I said here, we're going to get the insulin protein. Once the bacteria makes the insulin protein, it has to be purified. But in this process, by inserting genes into plasmids and putting them into bacteria, you can make bacteria produce any protein that you want it to be producing. Now, what we're going to identify now is that if you take a notice, we're cutting this DNA with this scissor, per se. We're going to now identify what this scissor is. It's not actually a scissor. And we're going to talk about these scissors actually have a name. They're called restriction enzymes. And sort of the next part of this lesson is what restriction enzymes do. And then when we get back from Christmas break, we're going to talk about how we use both plasmids and restriction enzymes to get genes that we want into bacteria. So again, this is really what this says here. When we get back from Christmas break, we're going to see how we can insert genes into plasmids. But right now we're just going to take a look at restriction enzymes and what restriction enzymes can do. So restriction enzymes, their actual name, or their fancy name, is restriction endonucleases. These were discovered in the 1960s, so even before my birth, and it's thought they were evolved in bacteria to cut up foreign DNA or to protect themselves from viruses, almost like siRNA was evolved. But it's thought that bacteria evolved these restriction endonucleases to cut up foreign DNA, like from viruses, when they got infected. And scientists actually found these enzymes in bacteria, and now we use them for our benefit. And not that this is that important, but some people wondered, or scientists wondered, well, how come the restriction endonucleases don't cut up their own DNA? Well, it was found that actually bacteria have the ability to methylate their DNA, which protects them from the restriction enzymes, and they also just don't have the base sequences that we're going to see in the second that are recognized by these restriction enzymes. Like, those sequences are not in their DNA. Okay, so basically, what do restriction enzymes do? Restriction enzymes cut when they see specific sequences, not just any sequence, specific sequences. And they I'm not going to make a big deal about this, but they cut at palindromic sequences that are the same frontwards and backwards. Palindrome is the thing, if you read it this way and you read it this way, it reads the same way. So if you take a look at this sequence here, there's a restriction enzyme that cuts anytime it sees the sequence in red. Okay? That site is called a restriction site, and they cut whenever they see this sequence, they'll cut the DNA using this pattern, in this pattern. Okay? And you'll see that when they cut in this pattern, that leaves something called sticky ends. Like we have this yellow sequence, right? And then we have this yellow strand that's been cut, and this blue strand that's been cut. And then we see it has a sticky end that overlaps here. So this AATT is a sticky end with this AATT. There are many of these enzymes and they all cut different sequences, or they all recognize different sequences and cut the DNA. So really, the point is they just don't go cutting the DNA all willy-nilly. They only cut when they can bind to the specific sequence and recognize that sequence, and they cut at that sequence in a particular pattern. To understand this a little more, we're going to watch this video or animation of what restriction endonucleases do.
Restriction endonucleases are enzymes that cleave DNA at specific nucleotide sequences. The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence GAATTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end a few nucleotides long, and the single-stranded ends of the two fragments are complementary to each other. These single-stranded ends can pair with each other, sticky ends. Once their ends have paired, the two fragments can be joined together with the enzyme DNA ligase, which reforms the phosphodiester bonds of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease can be joined together. Restriction endonucleases are fundamental tools in genetic engineering.
So what I want to point out here is that these two DNA samples, the purple one and the blue one, were both cut with the same restriction enzyme, EcoR1. And because of that, they have complementary sticky ends, these AATT, NTTAA. And because of that, if you put them together, they're going to anneal, and then they can be glued together with DNA ligase. So once again, restriction enzymes are enzymes that recognize specific DNA sites or specific DNA sequences and cut the DNA. And when they cut the DNA, they leave these ends, they cut in sort of like a zigzag pattern, and leave these ends called sticky ends. When we get back from break, we're going to do an activity that helps you sort of like realize what these sticky ends actually are. Okay? We're also going to see, as the sounds come back, that if you cut a gene that you want with a restriction enzyme, and then also cut, like, say, the plasmid that you want to add it to with the same restriction enzyme, they're going to have sticky ends that overlap. And then as a result, the gene that you want can be inserted into the plasmid, and then you can insert that plasmid into bacteria. Do not worry if you don't understand that now. The main purpose of this lesson was to realize what restriction enzymes do. And that's what this is showing. And this is what we're going to do when we get back from break. If you cut the human insulin with a restriction enzyme, and then cut a plasmid with the same restriction enzyme, they'll have complementary sticky ends, and they'll stick to each other. And then you can take that plasmid, which now has the human insulin gene, and insert it into bacteria, and then they'll produce human insulin. When we get back from vacation, we're going to be discussing that in greater detail. Alright, Kaiser's still sleeping. Have a good rest of break.